All right, class, here we go. Last chapter of the quarter. You've made it all the way through. I do want to point out that developmental biology is a very broad topic. People, get their degree PhDs in developmental biology, and so we've only really scratched the surface during this quarter. There's a lot of other things that we weren't able to get to just because of time constraints. So if you do feel that you want to dive deeper into this topic, the textbook or online resources, YouTube videos, etc., can kind of expand upon this. Evo Devo, evolutionary development, and things like that are additional topics that may interest you. But unfortunately, like I said, with the quarter structure, we're just not able to get to everything in this textbook. So with that, we are going to finish up with the endoderm. So how do these endoderm cells originate or arise? So there are some signals early on when the epiblast cells are becoming these intermediate mesendoderm cells. And so nodal WNT and BMP kind of cycle to create that conversion. And then depending on exposure to nodal here and to FGF or BMP, we have two different lineages we can go down. So if you have BMP and FGF, you become a mesoderm cell. If you're exposed to high concentrations of nodal, you become a definitive endoderm cell. Now, there is a gradient of BMPs, FGFs, and WNT here from the anterior to the posterior, and depending on how much of these markers you have, of these proteins you have different cell fate. So more anteriorly, you turn into an AFG cell and then into either lung or thyroid. If you're more in the middle of the anterior-posterior axis, you become a PFG cell and can either become a liver cell, bile duct, pancreas, or exocrine or endocrine pancreas. And then lastly, if you're towards the more posterior, you become an MGHG cell and you'll develop into intestine tissue. And just to elaborate a little more on here, the AFG stands for the anterior foregut. The PFG stands for posterior foregut, and MGHG stands for midgut or hindgut. So these two are obviously towards the foregut, so more higher towards the anterior end, and then midgut and hindgut obviously are more towards the posterior end. So initially, the endoderm cells form this flat sheet indicated as kind of gold here, surrounding this internal cavity of the developing embryo. And so if we were to take a cross section, you can see kind of looking down, this would be the notochord here and the developing neural tube. And then if we were to take a sagittal section or a midline section, you can see similarly this internal cavity with the developing endoderm cells. So as we proceed through development, we'll start to get more and more of this tube shape of the gut formation. So at this point up here, you can see that we have the cardiac primordia, so that is the beginning of the heart. So this is the anterior side here, and this is the posterior. And so we also have the allantoic diverticulum. I had to take it nice and slow to say that. Allantoic diverticulum, which is kind of the beginning of this caudal intestinal portal, which you see down here. So this is going to be the caudal or posterior side of the digestive tract. We also have the developing foregut and the anterior intestinal portal. So this is going to be towards the mouth of the digestive tract. We've developed the heart at this point, and we have a hindgut and a midgut. So we're starting to kind of stretch this out and create that tract that goes from your posterior to your mouth. Continuing on through development here at about 3 weeks, we see that there is chambers developing in the heart here. We're developing the pharynx on the anterior side, right? So up towards your throat here. We are developing the thyroid, also anteriorly. Right underneath the heart, we're starting to get these endoderm cells turning into liver tissue. We're starting down that specification to liver tissue. At about 4 weeks, as you can see, we're continuing on to develop these organs that are surrounding the digestive tract here. We have the pancreas now starting to emerge, along with further development of the liver underneath the heart that is developing those chambers. The stomach is starting to develop anteriorly to the yolk sac, and we're getting lung buds right next to the heart there, in addition to the thyroid and the beginning of the mouth. And additionally, you can see here at the very anterior part of the developing of the neural plate / neural tube, the brain is forming, so you're getting a big bundle of nerve tissues there. And then caudally, or posteriorly, we see that we're starting to develop the anus, and you can really see, let me change my color, the development of this digestive tract here, where this is going to be the intestine, stomach, all the way up to the mouth. So the anterior of the endoderm of the digestive and respiratory tubes begins at the pharynx. So during development, the mouth is divided into 4 pairs of these pharyngeal pouches. The teeth and the major salivary glands are from the ectoderm, and the anterior taste buds are from the ectoderm, but the posterior taste buds, as well as the salivary and mucus glands in the back of your mouth, are from the endoderm. So as I said, there are 4 pairs of these pharyngeal pouches, and only one side is shown here, so it's symmetrical. So you'd have another 1 and another 2, 3, 4, etc. on this side. The end of each of the first pharyngeal pouches becomes the tympanic cavity in the middle ear. The second pouch receives lymphoid tissue, and it will become the tonsils. So tonsils is part of your lymphatic system. So when you are sick, your tonsils swell. The dorsal portion of the third pharyngeal pouch forms part of the parathyroid gland, and the ventral portion forms the thymus. And both of these will migrate caudally and meet with the tissue from the fourth pharyngeal pouch. As you can see here, they're migrating. That forms the rest of the parathyroid and the postbrachial body. In addition, the thyroid, which we can see over here and forming right here, that will also migrate, and it will move further down caudally or posteriorly into the neck region as the embryo develops. So initially, this digestive tract forms as a tube, but then we need to specify it, right? Because our digestive tract isn't just one long tube. There's organs within it, such as the stomach, the large intestine, the small intestine, the rectum, and things like that. And so the regional specification of the endoderm and the splanchnic mesoderm occurs through these specified genes. And so SOX17 is a gene that is expressed throughout the entire digestive tube. So that's kind of one of those genes that says, let's become the digestive tract, but it doesn't give polarity to it. And so it's just kind of, all right, let's make all this digestive tract, then we'll specify later. And so then we have another SOX gene, C-SOX2, which will specify the development of the esophagus and the stomach. PDX1 will be expressed more caudally, and it will lead to the development of the intestine. We have the HOX genes that will help to specify the liver. CDXC will lead to the development of the small intestine in addition to PDX1, so the more anterior portion of the small intestine. CDXA will help to develop the large intestine, colon, and the rectum. So how are these different genes activated or expressed in different tissues? And so the digestive tube passes through a large part of the developing embryo, and as that tube encounters different mesenchymes, these mesenchymes instruct the endoderm to differentiate into different tubes. And so, for instance, in the stomach, bar X here activates these SFRPs, which block WNT and causes that tissue to differentiate into stomach tissue. In the intestines, because it's experiencing different mesenchyme, there is no bar X, and so there's nothing to block WNT, so we'll put a plus here because we're good to go, and thus we develop into intestinal epithelium tissue. Now, more caudally, we see that from the stomach at least, we see that there is another gene that isn't on here, CMP4, which is expressed by the surrounding tissue, and that instructs the anterior mesoderm to express SOX9 and NKX2. And these are transcription factors that lead to the expression of other genes that develops the sphincter muscles. So the mesoderm tissue surrounding the endoderm continues to play a role throughout development. And so they interact through this dorsal mesentery. And so this connection between the mesoderm and the endoderm, there's this fibrous membrane that connects the two. And that is partially what is responsible for the folding and kind of bundling of the intestines, you know. So if you've taken anatomy or just any general biology course, the intestines are all folded up and kind of spun up inside your body. Well, if you sever that connection, the gut becomes this long, thin tube and it loses its shape. However, if you do have a proper interaction between the mesoderm and the endoderm, you get the proper kind of folding and secondary structure that's present in these developing gastrointestinal tissues. So now we're going to take a look at some of these kind of accessory organs to the GI tract, and one such is the pancreas. And so the pancreas, if you've taken anatomy, you may know it's lobed. So there's kind of a anterior and posterior lobing to it, and this is the result of fusion of a dorsal and a ventricular diverticula. And so as they grow, they come together and eventually fuse. So if we look at day 30 over here, we see that we have this ventral bud and this dorsal bud. And by day 35, there's the beginning of this migration that the ventral pancreas at this point, or what was the ventral bud, will move towards the dorsal bud. And so by day 40, you can see that they are starting the process of fusing, and we have the bile duct that is interacting with the GI tract. And then by the 42nd day, we have fusion, and the ducts will no longer, there won't be multiple ducts like there was at day 40, and you'll lose a duct, and there'll only be one duct interacting with the GI tract and in the duodenum. However, in about 10% of the population, there is still this dual duct system where each of the two previous buds will still interact independently with the GI tract. So we're going to continue on with these accessory organs and talk about the development of the liver. And so the liver tissue will only form when it is near cardiac mesoderm tissue. So it only forms underneath or more posteriorly or caudally to the heart. And this is likely due, it's not 100% certain, but it's likely due to FGFs that are secreted by the heart endothelial cells. And so the ectoderm and the notochord block the ability of the endoderm to express liver-specified genes. So as you can see here, the ectoderm and the notochord are repressing the genes that would allow for liver formation. The cardiogenic mesoderm, likely through FGF1 or FGF2, promotes liver-specific gene transcription by blocking the inhibitory factors that the ectoderm and the notochord were sending out. And so this will allow those genes to be expressed and allow for this region right here of the endoderm near the cardiac tissue to eventually develop into liver tissue. So the formation of the pancreas is kind of opposite of the liver. And so in the pancreas, the heart cells prevent formation of the pancreas, and the notochord promotes it. While in the liver, it's the opposite, where the heart cells promote the formation of the liver and the notochord prevents it. And so here we have, we're away from the heart cells. We're on the opposite side of where the heart forms. And as you can see here, there is a gene, PDX1, that the gut borders the aorta and these vitelline veins. And where the gut kind of interacts or comes close or in contact with either of the circulatory structures, there is induction of this PDX gene, which causes the formation of those buds for pancreas formation. Now, the previous slide had two vitelline veins, and that was where the example of a chick embryo, which forms those two buds on the ventral side. But in the mouse, only one of these veins survives, and thus the induction of PDX causes budding of a dorsal and ventral bud, but only one in contact or near that vitelline vein. And note that we have no heart tissue, mesoderm that's going to become heart tissue nearby, and thus we are able to develop into pancreas as opposed to liver. So here we have the digestive tube as indicated with my green squiggly here in the middle. And this is a stain of mRNA, so the precursor to the protein for PDX1. And as you can see, I will mark the veins here. We have the two veins, the aorta and the vitelline veins. And in the regions that are nearest the two vitelline veins and the aorta, which is up at the top here, in those regions nearest there is where we have expression of this PDX1. So in order to become pancreatic tissue, you have to be near these vitelline veins or the aorta because those paracrine factors are needed to cause that differentiation. So the pancreas isn't only there for secreting digestive enzymes, it's also got endocrine properties. And so these are generated by the islets, which are specialized cells within the pancreas that create insulin and things of that nature. And so the way that these islet cells differentiate is through interaction with blood cells, or blood vessels, excuse me. And as you can see in this fluorescently stained image here, the red indicates blood vessels that are interacting and causing the development of these islets, which are stained green here. And the blue stains is DAPI, which is a stain for the nucleus. So taking all these things together, we can kind of map this out. So we have the ventral endoderm, which is near the cardiac tissue and kind of not as near or not as influenced by the notochord. And we have the dorsal endoderm, which is away from the cardiac tissue and more present or closer to the notochord. And so if we're in the ventral endoderm, there are a few genes here, FGFs, BMPs, FOXA1 and FOXA2, that leads to the formation of hepatoblasts, which are these undifferentiated liver cells. And then the presence of a couple more genes, HNF6 and OC1, will lead to bile duct formation, while HNF4 leads to hepatocyte or liver tissue formation. And if you are on the other side, if you are away from that cardiac tissue, instead FOXA1, FOXA2, HTXB9, and PTFTA will lead to the formation of pancreatic buds. And these pancreatic buds will then form into either exocrine progenitor cells that will lead to the formation of ducts or the acinar cells, or conversely, if you are going to interact with blood vessels, this NGN3 and FGF10 will lead to these endocrine progenitor cells. And through a cascade of different genes here that I'm not going to name them all, you can either become beta cells, alpha cells, PP cells, etc. So knowing this fate mapping of the specification of cells such as pancreatic cells leads to the possibility of using them for therapeutic purposes. And so we kind of briefly hit on this in chapter 5, but what scientists have been able to do, and hopefully in the future we'll be able to do in humans, is take an adult skin cell and using the transcription factors that we know about, revert that to a induced pluripotent stem cell. And because we know this fate mapping in pancreatic cells, we can mimic the environment and the specific differentiation factors that lead to pancreatic cells and hopefully create these patient-derived, so to speak, cells and implant them back in and allow that to make up for the cells that are not capable of producing insulin. So in this example here, researchers took these stem cells, or skin cells, excuse me, created IPS cells, or induced pluripotent stem cells, and then provided it SOC17 to make definitive endoderm. From there, the addition of FOXA2 and HNF1B created primitive foregut. Expression of PDX1 then leads to these pancreatic progenitor cells, which will develop into endocrine progenitor cells, and then with the expression of NGN3, we create beta cells. And these beta cells then, which are producing insulin and releasing them into the environment, we can then transfect or put them into a mouse that was deficient in these cells before and was unable to produce glucose when to metabolize glucose in fasting. We're able to restore that function in the mouse with these induced pluripotent stem cell derived beta cells compared to a diabetic mouse that had no ability to process this glucose when fasting. So when you hear of patient-derived stem cells and the idea of tailoring these to cure diseases, this is an example. And fate mapping is a very important part of that because we need to know what these intermediate factors, such as NANOG and SOC17, etc., that are required to move something from being a pluripotent stem cell to an end product like a beta cell. So briefly, we're going to hit on a little bit of interesting cases with the gallbladder here. So there's this phenomenon or this dysfunction called biliary atresia. And in some mammals, including people, they're born with clogged bile ducts, which is called biliary atresia. However, it's starting to appear that environmental factors during development can play a role in this. And so one case study was done with cattle in Australia. And so these cattle were having an increased prevalence of this blocked bile duct because of drought. And so it didn't make sense that because there's just lack of water that they would have this malformed gallbladder. And it turned out that it was because that during drought, a certain plant became more prevalent because it was able to outcompete the rest of the native plants when there was low water conditions. And it came to light that these cattle were eating a plant that was producing a compound called bilatestrone, bilatresone, that was from this plant called pigweed, and it would alter the lumen in these cells. And so during development, if you were to ingest this compound, this bilatresone, during this developmental process, the bile ducts associated with the gallbladder were then closed. And so you'd have this kind of environmental developmental interaction that occurs. So although it doesn't appear intuitive, the respiratory system and the lungs and the trachea are actually derived from the digestive tract. And so this is the partitioning of the foregut into the esophagus and respiratory tracts, which happens during the 3rd and 4th weeks of human gestation. And so here we see kind of a lateral view of this respiratory diverticulum, and we see a ventral view in this case here. So as you can see, the developing foregut from the endoderm here starts to partition at 3 to 5 weeks, where we're going to seal this off to make the trachea and the esophagus. And eventually you have 2 independent tubes with the pharynx at the very top of the esophagus and the trachea, and then the eventual beginnings, at least, of the lungs, starting with these lung buds. So just like everything else we've talked about, there seems to be a few genes involved in this, and these paracrine factors repress or let expression of these Hox genes or developmental genes to occur. And so in this case here, we have mesenchyme that is around the esophagus that expresses this BARX1. And because of that, there is a blocking of WNT, which results in the tissue of the digestive tract developing into esophageal epithelia. Now, a little bit more ventrally, if there is no expression of BARX1, this allows WNT to not be blocked, to continue its expression, and that results in the development of the digestive tract to become the ciliated respiratory epithelia. So that becomes your trachea as opposed to your esophagus. So we're going to finish up here by talking about this very cool system of signaling for contractions for birth. And so in the embryonic lung, once it's to a certain developmental stage, that triggers the start of the birthing process. And so in this case, we have surfactant A that activates these macrophages in the amniotic fluid from the lungs. So the lungs exposed to the amniotic fluid as it gets developed to that point, and it
because obviously the fetus is ingesting this amniotic fluid, and that's why after birth they have to like get the fluid out of the lungs of the baby for the baby to breathe. But anyways, this surfactant is exposed to or transferred to the amniotic fluid, which activates these macrophages. And these macrophages, in turn, will stimulate the production of this interleukin-1 beta, or IL-1 beta, which stimulates the production of cyclooxygenase 2. And in the presence of arachidonic acid, excuse me, there is prostaglandins that are activated, and unfortunately under my picture here, that leads to is taken up by the uterus, or the signal is received by the uterus of the mother holding the child, and that leads to these uterine contractions. So this is one method of ensuring that it's not until the lungs are at a certain developmental stage that the baby is ready to be born. Because if those lungs aren't sending the message that, yes, I'm ready to take an oxygen myself, I don't need the umbilical cord anymore, then you would give rise to premature births and babies that wouldn't be able to survive. But this is kind of a safeguard to say, okay, the lungs are developed enough, we're going to use this macrophage-derived pathway to send a message to the uterus to say, okay, it's time to give birth.